A forensic scientist sits in her lab with 3 DNA samples in front of her. One sample is the DNA left behind at the crime scene by the criminal. The other 2 samples are DNA from possible suspects. How will she know whose is whose? The scientist knows she can use an enzyme to cut each DNA sample at a particular sequence of nucleotides. This will leave behind several different pieces of DNA. The exact number of pieces and their sizes will be unique to each individual. This means that there will be an exact match in the pattern of different sized pieces of DNA between one of the suspects and the DNA left at the crime scene, but not to any other suspect's DNA. The only problem left is, how will she see and measure the different pieces of DNA in each sample? The answer is gel electrophoresis. Gel electrophoresis is a technique used to separate and view macromolecules. Macromolecules are large molecules such as DNA, RNA, and proteins. During gel electrophoresis, the macromolecules are loaded into a gel. Then a current is applied across the gel. The result is a separation of the macromolecules based on mass. In order to see the macromolecules in the gel, scientists add either dyes, which stain the area of the gel that contains the macromolecules, or chemicals that bind the macromolecules and fluoresce when the gel is exposed to ultraviolet light.
So how does gel electrophoresis work? It is based on the principle that nucleic acids, like DNA and RNA, are negatively charged. This means that if you put nucleic acids in an electric field, they will migrate away from the negative end of the field and toward the positive end. The nucleic acids are placed inside the gel for 2 main reasons. 1. The gel is a way of holding them to know where they are. 2. The migration needs to occur in a manner that allows for the separation of different sized pieces of DNA or RNA. The gel has many microscopic holes through which the nucleic acids wiggle as they migrate within the electric field. The smaller the nucleic acid sequence, the easier it is for it to wiggle through the holes. So, smaller pieces of DNA and RNA run through the gel faster than larger pieces. Returning to our forensic science example, this means that the individual pieces of DNA in each sample are sorted within the gel. The larger pieces appear at the top of the chamber and the smaller pieces appear at the bottom of the chamber. The scientist compares the pattern of the pieces of the crime scene DNA to the pattern of the suspect's DNA and looks to see if there is an exact match.
When biologists want to separate different pieces of DNA, RNA, or proteins, they use a technique called gel electrophoresis. In this science project, we'll build a gel electrophoresis chamber and use it to discover how many components of food coloring dye are in mm's. Agarose gel electrophoresis has proven to be an efficient and effective way of separating nucleic acids. Agarose's high gel strength allows for the handling of low-percentage geles to separate large DNA fragments. We use distilled water because it does not contain impurities like dissolved ions. A sodium bicarbonate solution will function as the electrolyte, which will channel the electric field through the gel. It will be used as an ingredient in creating the gel, and it will also surround the gel during the electrophoresis. Seed round: a rectangular plastic container, 5 9-volt batteries, 2 alligator clip wires, wire, and a microwavable bowl, and of course, M&Ms. The first step in the experiment is to make the buffer solution you will use for making the agar gel and running the samples. The buffer should be a 1% solution of baking soda. To make this, stir well by mixing 2 grams of baking soda with 200 milliliters of deionized water in one of your bowls. Then make a 1% agar gel solution by combining 1 gram of agar powder with 100 milliliters of your buffer solution in a microwave-safe bowl. Heat the agar solution in a microwave to dissolve the powder. Gently pour the agar solution into the gel chamber. Wait until the gel solidifies, which may take at least 30 minutes at room temperature. And it's still liquid. What happened? Heat weakens chemical bonds by making molecules easier to dissociate. You can think of it this way: chemical bonds are a form of negative energy. If you add energy, the energy is less negative. When the energy goes to zero, the molecule breaks apart. So I reevaluated and added more agar and stirred it every 10-15 seconds while it was in the microwave. Because what do you call an experiment that works the first time? Boring. Pour the remaining 100 ml of your buffer solution over the solidified gel. Using the alligator clip leads, attach the battery pack to the wires resting on the gel chamber. The positive terminal of the battery pack should be connected to the positive electrode. This is the electrode toward which you want the food coloring dye to migrate as it separates. You should see bubbles forming around the electrodes in the buffer as the current passes through them.